Okay, good morning everyone and welcome to the 2016 Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present a familiar face, Dr. Annie Quinney. Annie is somewhat of a poster child for the Royal Tyrrell Museum, as she grew up in Drumheller and worked at the museum for many summers during her high school and early university years. Her experience at the museum sparked her interest in geology and paleontology, leading her to pursue a bachelor's degree in geoscience at the University of Calgary. As an undergraduate student, Annie was heavily involved in scientific research projects, obtained several research grants, and completed an honors thesis on the geomechanics of living and extinct mammalian predators, including the saber-toothed Smilodon. After obtaining her bachelor's degree, Annie decided to stay at the U of C to pursue a master's degree, for which she studied the ancient soils of the Horseshoe Canyon Formation exposed here in the Drumheller Valley, in order to reconstruct the ancient environments and climate in which dinosaurs lived 72 million years ago. After successfully completing her master's degree, Annie moved to the land down under to pursue a PhD in the School of Geoscience at Monash University in Melbourne, Australia. For her dissertation, Annie studied 90 million-year-old amber pieces from Australia and Canada in order to search for potential inclusions and to reconstruct high-latitude environments and climate during the early late Cretaceous. After completing her PhD in 2015, Annie was hired as a postdoctoral fellow at the Arctic Institute of the University of Calgary, where she leads many public education and outreach programs about the consequences of modern climate change and Northern Lights as well. Today, Annie will discuss the results of her research on Cretaceous amber from Australia and Canada and present an overview of her work at the Arctic Institute. So without further delay, I present you Dr. Annie Quinney.
Thank you, Francois. I have to apologize. I'm totally losing my voice, so I'm going to speak very low. It's going to be very Maryland up here today. And I was promised a fan, but I never got it. So anyways, that's okay. We'll make do. And I'm just laughing because of Francois's intro, and I was remembering there was a time when we used to have to sit out at the hoodoos to protect them. That was my job when I was, I think, first year undergraduate. And Francois came out and was showing me cool things in the rocks at the time. I think you were actually trying to suck me into paleo. That was like way back when. And he did. I ended up doing my master's with Francois, and he helped with my PhD as well. So you did well. Sucked me right in. It's really nice to be back here today. I guess it's been a few years since I did my last talk for Speaker Series. But yeah, since then I've done a couple of new things, and that's what I'm going to be talking about today. My PhD research, which was on amber. And it was a bit of an adventure. But anyways, you'll see that as we go. So this was the research that I did for my master's. And when I finished my master's degree, I was really full of optimism and hope and energy for the future. And I was ready to take on the world as a young paleontologist. But not surprisingly, I spent the next several months unemployed, just waiting for a job to come to me. It definitely did not. And that's when I decided that I needed to go back and get a PhD. So I started trolling the internet and calling around and seeing what I could find. And I ended up calling Monash University in Australia, of all places. And I'm totally paraphrasing the conversation here, but this is kind of how it went. Hello, Monash University, it's Annie, as if they know me. I'm totally unemployed. Do you have any cool projects? And when I told them I was looking to do paleo, they were much more receptive to this conversation. And they replied, yes, we absolutely do, but it is top secret. I'm not kidding you. They told me it was top secret. They had found the first piece of Mesozoic amber from Australia. And what you need to know is that there are a couple of other deposits of amber in Australia, but they're quite a bit younger. They're Miocene and Eocene in age. And actually one of them isn't even Australian. It floated from Southeast Asia across the ocean and just landed on the beach in Australia. But they claim it as their own. So this was kind of a big deal. It was old and it was new and it was interesting. And that's a picture of the piece of amber that they found. It's less than a centimeter, just so you know. It's not very big. And so I thought, okay, that's pretty interesting. But Monash, how do I know that you're the right university for me? To which they replied, well, Annie, we have money. So I thought about it for a few seconds, and I was like, I'll be right there. I'm on my way. Literally, I hopped on the next plane to Australia. And if you've actually done the flight to Australia, you know that this diagram is a complete and utter lie. It's actually 3 to 4 layovers and 24 to 48 hours of your time wedged in between either a football player and a cuddler or a drooler, right? You're just wedged in there. So luckily, my flight was only 40 hours. Thank goodness. And I landed in Melbourne to start my PhD adventure. So once I got there, more information about this really exciting amber deposit started trickling out. And my supervisor at the time, he told me that the amber that he found came from a petroleum core that was drilled in the Otway Basin, which is just off the southern coast of Melbourne. And there were a bunch of wells that were drilled in the area at the time. They weren't that fruitful, but anyways, they were drilled. And this is just a small representation of some of the wells that were there. And he was just totally convinced there was going to be tons and tons of amber for me to study. So I had a big project ahead of me. And so I thought, sweet as. Now this is a little bit of Australian lingo that I picked up while I was there. And I hadn't seen it written down, so for the longest time, I actually thought people were just complimenting my bum. But then I saw it written down, and actually it just means neat. So neat, I was going to have lots of stuff to work on. But then the story got even better. This wasn't the end. They thought they might have a mite in the amber. And I was kind of like, a mite? All right, whatever. But they were like, a mite. This is a really big deal. And to understand why it is a really big deal is because Australia doesn't actually have that many fossils. And the reason for that is because Australia is really, really flat. So there aren't a lot of mountains or valleys or canyons that expose the rocks, that expose the fossils. So actually having a fossil in amber was pretty exciting. What's even more interesting to me, though, is the fact that during the time when the amber was deposited, which was 90 million years ago during the mid-Cretaceous, Australia was still connected to Antarctica as part of the supercontinent of Gondwana. And this kept Australia much further to the south, so much so that the southern margin of Victoria, which is in that red box there, was actually within the Antarctic Circle. So here we had a piece of amber that told us that there was a thriving resin-producing forest within the Antarctic Circle 90 million years ago, and it may have had a mite in it. Pretty exciting. So I went back and started looking through those wells, and I think, if I remember correctly, I looked at about 50 different wells, 50 different cores from 50 different wells. And we did find amber in them, but not in that many wells. I think I collected from 6 of those wells in the Otway Basin. And we did collect about 200 fragments of amber. And they looked like this. So you can see that there's a big range of color. There's yellow, there's orange, dark orange, red, and brown. It doesn't show up great on here, but there was a big range of color. And I looked at every single one of these pieces of amber under the microscope because I was hoping for, you know, a mite or a fly or a mosquito. Anything would have been good. Instead of that, I got this. No bugs at all. Sort of planty material is what I ended up with. I had a couple of spores, but most of the plant material is actually really degraded, so you can't actually tell much from it because it's broken down. And we also had these sort of stringy creatures in the amber, which were some kind of filamentous organism, either fungal hyphae or some kind of bacteria. So that was the excitement in my amber. 2 spores and some dirt, really. In addition to those inclusions, there were these other funny inclusions in the amber that came in a bunch of different shapes. So there were circles and ovals and more stringy-like things. And they looked a little bit strange. They kind of looked like some modern organisms. So I've done a little side-by-side here. The first column is what I have in my amber, and then what I thought they looked like, their modern representative, is in the second column. So just so you can see like what I was thinking here, they do look pretty similar, right? But when you actually start looking carefully, you start to notice that these things don't quite match up. Most importantly, size-wise. So if you look at the scale bars here, the ones that were in the amber were either way bigger or way smaller than their modern representatives. There were also irregularities in form and missing diagnostic structures. Like, what was so strange was that usually in paleontology you have the hard parts preserved, the robust things, but here we seem to be preserving only the delicate things, and there's really no reason for that to happen in amber. So what we ended up deciding was that these were fossil imposters. They were not fossils at all. Somebody else had actually done a study on this as well, and he coined the term pseudo-inclusion, which basically means fossil look-alike. So they're actually just structures that form in the resin as it's coming out of the tree. So this made me question, A, my degree, and B, my amber. So I took another look back at the mite. So this is a picture of a modern mite here, and that's the picture in the amber. And you'll notice in the modern mite, it has 4 legs on one side, it has 4 legs on the other side, and it has a body. In the piece on the amber, it has 5 legs on one side, 1 leg on the other side, and no body whatsoever. That's a bit of a weird mite. Additionally, its legs seem to be broken in multiple places, and whereas the legs on modern mite sort of taper towards the foot, these legs seem to vary in width with each segment. Questionable. Very questionable. But we were still so excited about the prospect of having a mite in Australian amber that we decided we still needed to do some more tests. So we ended up scanning this piece of amber with multiple different instruments. We did synchrotron, CT, ultramicroscope, you name it, we did it. And the results were all the same. It was empty. Uh-oh. So for these scans, they really rely on density contrast, and because mites have an exoskeleton that is quite dense, it should have shown up in the image. So had there been a mite there, it would be in this picture, but it's not. All we're seeing are flecks of dust. No mite to be had. So what's a paleontology student with no fossils to do? Come up with another plan. So we ended up spending a lot of time looking at the pseudo-inclusions to see if we could come up with any sort of, relationship between amber and the environment, or if they told us anything about how the amber formed. And we did find a couple of things. We noticed that some of the pseudo-inclusions were just sort of randomly oriented in the amber, and others were mutually aligned side by side. And the ones that are mutually aligned, that's evidence of flow, because these, as I mentioned, these are structures that are forming as the resin is sort of oozing out of the tree. So if that resin is flowing, it's going to cause those structures to become aligned. But if that resin is not flowing, they can just stay randomly oriented. So if they're randomly oriented, we interpret that resin has hardened in place right where it came out of the tree. But if they're mutually aligned, then that resin has sort of dripped down the tree. So evidence of movement. Okay, the other type of inclusion that we have in the amber is dust. It's a really common inclusion, and if you've ever been in a dust storm, you know that dust is really common close to the ground, but as you get further up, it sort of dissipates. The wind can't carry it too high above the ground. So we had this idea that perhaps amber that forms, or resin that comes out of the tree sort of lower to the ground, might be more full of dust than resin that comes out higher up in the tree where it's out of the range of where wind transported dust could be. So those were our big ideas. Then we decided we were going to compare the entire range of inclusions, dust, pseudo-inclusions, and our very exciting biological inclusions, to color. And we kind of came up with a cool relationship. So the yellow amber is typically clean and clear. It doesn't have any inclusions in it at all. No dust, no pseudo-inclusions, no bio-inclusions. Occasionally, it will have one of those randomly oriented pseudo-inclusions, and so that suggests to us that yellow amber probably was resin that seeped out of the tree and hardened right in place, right where it came out. Now the orange and dark orange ambers, they had pseudo-inclusions that suggested they could have either stayed in place, hardened in place, or dripped down the tree. Either or, they had both, and both were common. And they were also full of dust. Red ambers, like the yellow ambers, also had those randomly oriented pseudo-inclusions, so they probably hardened in place. But unlike the yellow ambers, they did have a bunch of dust in them. And then the brown amber, which was a little weird, was opaque. It was so loaded with dust and debris and plant fragments that we couldn't actually see through it to see if there were any pseudo-inclusions. So this was kind of a neat piece of information. It told us that each color of amber had a specific association of inclusions, and we decided that we could use this to predict where on a tree that amber formed. So that clean, clear yellow amber is probably coming from pretty high up in the tree, where it's out of the range of the wind because it's dust-free. And then our dust-filled orange, dark orange, and red ambers are probably lower down on the tree trunk, and they're just being completely washed with dust. And then our brown amber, even though we couldn't see any pseudo-inclusions, it's probable that it dripped off the tree, landed on the soil, and engulfed fragments of dust and plants that were rotting at the surface. And that's why it's so loaded with debris. So this was a pretty cool story, and it actually did end up getting published, but it did not a thesis make. We had more stuff to do. And I ended up deciding that I needed to expand my field area. So I've got my Otway Amber location down there. This is what the Cretaceous world looked like. And I decided I needed to go look for 90-million-year-old amber in the northern hemisphere so that I could do a comparison. And I was hoping that somebody had already found that amber and it would be really easy for me, but of course that didn't happen. So I had to sort of look through the literature to find rocks that were 90 million years old that had formed on land so that I could go look for amber. And where did I end up? Back in Canada. Hooray! It's a good place to be. I actually ended up back in northern BC and northern Alberta near the towns of Tumbler Ridge and Dunvegan. And I've got a couple of field photos for you. So they're actually beautiful field localities. They were really nice to work in. That's Quality Creek right there, which is near the town of Tumbler Ridge. And if you're at all familiar with it, it has a lot of dinosaur footprints along it, which were really cool. I totally wanted to steal them, but that was totally unethical, so I didn't. And then Dunvegan is just up from the bridge. And you can see that we had some success. We were able to find some amber. Most of the amber I collected came from the Dunvegan locality, which is actually just a little bit older than the Australian stuff. But we did find a few pieces from the Quality Creek, which was perfect because then we could do a comparison. So after I collected my amber, I did scan it for inclusions, and it turns out that the Canadian amber really didn't have much in the way of inclusions either. But it turns out that's actually pretty common. Inclusions are more rare than we would like to think. But anyways, so it was pretty devoid of inclusions. So my next step was, why not do some chemistry? Why not see if there's some differences between the Canadian amber and the Australian amber? So the first chemical analysis I did was FTIR, and you don't need to know what that is. All you need to know is that it tells you what types of trees were producing that resin. And it turns out that every single amber piece that I collected was produced by the same group of trees. And that group of trees includes the Araucarians, if you're familiar with those. So if you were thinking to my color story and wondering if you could attribute the different colors to different types of trees, I beat you to it. That's not the case. They were all produced by the same type of tree. But this was actually a really good result for us because it meant that in other chemical analysis, we could actually compare the results directly, which was perfect. So our next chemical analysis was stable isotopes, and I'm just going to talk about carbon isotopes today. So we know that trees suck up carbon dioxide from the atmosphere, but not every single molecule of that carbon dioxide is created equally. It can have different weights of carbon in it. So there's two different weights of carbon that we're concerned with. There's heavy carbon, which is carbon-13, and there's light carbon, which is carbon-12. And I put together a great diagram for you. Yes. So trees really prefer to use the light carbon because it takes less energy to use it for metabolic processes. So they're going to choose more light carbon than they are heavy carbon, and that's called fractionation. And when it comes to actually producing resin, to the manufacturing process of resin, the same thing goes. That tree is choosing the light carbon over the heavy carbon because it's easier to use. Now, the actual proportion of heavy to light carbon that that tree uses depends on a bunch of factors. It depends on the proportion of heavy to light carbon that's actually in the air, the concentration of oxygen versus carbon that's in the air. Regionally, it depends on precipitation. And then locally, it depends on water availability, composition, humidity, temperature, blah, 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 blah. A whole bunch of things can affect the ratio of how much heavy versus how much light carbon we're using in the manufacturing process. So I did the analysis for the Canadian and the Australian ambers, and this is the result I got. You don't really need to worry too much about the notation. All you need to look at are the numbers. And you can see that they are very, very similar. So we're looking at amber that came from the northern hemisphere versus amber that came from the southern hemisphere, and the numbers are almost identical, which is pretty cool. So the next thing I decided to do was to compare my high latitude amber from the northern and southern hemispheres to amber from the mid latitudes. And lucky for me, I didn't have to go searching for this one. It was already found. There's a 90 million year old amber deposit from New Jersey that had already been analyzed. I didn't even have to do any more chemistry. All I had to do was open a book. It was so great. And the results that they got in their analysis was negative 22 per mil compared to my negative 24 per mil. So this is a significant difference. And what it tells us is that Canadian and Australian ambers contain more light carbon than the New Jersey amber. So high latitude ambers have more light carbon. And trees at high latitudes have a stronger preference for light carbon. But the question remains, why? But when we're thinking about what the difference is between mid and high latitudes, there's two big factors that are very, very different. The first one, of course, is temperature. It's colder the farther north you go. It's warmer at lower latitudes. And the second one is light. So in the high latitudes during the summer, trees are getting exposed to, well, trees then, were getting exposed to 24 hours of continuous low-intensity daylight. But in the mid-latitudes, it was more diurnal, so sort of half day, half night. And this has a big impact on, has been shown to have a big impact on this fractionation, this selective process. Now, there was actually a study done by Ralph Tappert, and he was able to show that unlike other isotopes of like oxygen, for example, temperature actually has very little to do with fractionation in trees. So temperature is not really what we're looking at here. Actually, what it ends up being the controlling factor is the partial pressure of oxygen in the atmosphere. But anyways, all you need to know is that it's not temperature, and that leaves us with light. Light is the big controlling factor here. So there's a big discrepancy between low and mid-latitude ambers and high-latitude ambers in terms of their isotopic composition because of light. So that was sort of pieces of my PhD thesis. Currently, I'm working for the Arctic Institute of North America, and my job there is to develop their educational programming. The Arctic Institute of North America has been around since 1945, and so last year was our 75th anniversary, and during that time, we adopted this vision of advancing knowledge for a changing North because, as we all know, climate change is amplified in the Arctic. It's 2 to 3 times more intense in the Arctic. So that's sort of what we're working on. And a big part of that is outreach. So we run a speaker series and we have classroom programs. By we, I mean classroom programs is me coming to your classroom with some artifacts and talking to grade 2s, which is really fun. But anyways, we also have an Arctic journal, which is available online, and it's totally multidisciplinary. So anything from biology to geology to sociology, archaeology, it's all featured in that journal, and it's been around for a really long time. Specifically, some of my projects include, this is one of the big projects that I'm working on. It's called Polar Voices, and this is a podcast on climate change in the Arctic and the Antarctic. But it's climate change from both the perspective of the researchers and the perspective of Indigenous people that are actually living in these places and being affected by climate change. And I keep trying to suck paleo into this program. We've done a couple of episodes on permafrost, and I think we should really talk about creatures that are coming out of the permafrost. But I haven't won that battle yet. I'm working on it, but I will get there. Anyways, if you're interested in the program, it's available at thepolarhub.org. The next big project I'm working on, I'll just let this video run and then I'll explain. But this is some footage that was captured, this by the Canadian Space Agency, and then some photos of an array of cameras that we have across the Canadian North. It's about a minute long. Anyways, so we've kind of partnered with the Canadian Space Agency and with the Department of Physics and Astronomy at the University of Calgary, AuroraMax and Aurorasaurus, to come up with this massive citizen science project. And this is just a sort of a screenshot of our webpage. And basically, the physics department has a big array of cameras across the Canadian North that are just sitting there taking a picture every 10 seconds. So we have tens of millions of photographs that nobody has looked at. So we've taken this huge database of photos and we've put it online. And we're asking people to come and help classify the types of auroras that are in those photographs. So what you do is when you get to the main page, you hit on the tutorial, because if you're not familiar with the different types of aurora, you can't do the classification. So the tutorial will sort of run you through what a single arc is, what a multiple arc is, patchy, diffuse, so on and so on. And then once you feel like you have a pretty good sense of what those different types of aurora are, then you can go into the Classify tab. And so here, you'll be shown a picture from one of those cameras, and it will tell you where that camera is and when that photograph was taken. And then you just go to the panel up here and you select the ones that you think that this image represents. So this one's kind of confusing, but there might be an arc and it might be diffuse or it might be something else. If there's no aurora at all, which happens also, you can say, I don't see an aurora. Or if you just don't have a clue, you can say, I don't know. The really good thing about this project is that you're not the only one who's going to see that picture. Tons and tons of other people are going to see that image, and then we're going to use statistics to sort of figure out what we think the average response is. And if 98% of people agree that, yes, this is a single arc, then that gets right into the database, classified. But if it is 50-50 and we're just not sure, then the researchers can go in and look at that image and come up with an interpretation themselves. Anyways, all the classifications that get done by the public, they go directly back to the researchers to actually study the physics of the aurora. So your contribution is actually contributing to real science, which is very cool. Here are a few references from the PhD work that I did. Thank you so much for coming. I'm going to cut myself off because I just can't talk anymore. Thank you very much.